Regenerative medicine is emerging as one of the most tangible and revolutionary domains in anti-aging science, offering the possibility not just of slowing aging, but of physically repairing the damage it causes. As our understanding of cellular biology, tissue engineering, and stem cell differentiation deepens, the prospect of rebuilding age-damaged organs from within is moving from distant ambition to clinical reality. Instead of relying on invasive transplants or managing symptoms through lifelong medication, future medicine will likely involve regenerating tissues with patient-specific cells, restoring function and resilience in ways previously thought impossible. One of the clearest signals of this shift comes from advances in cardiac regeneration. Scientists have successfully grown patches of human heart muscle from induced pluripotent stem cells, iPSCs, cells that are reprogrammed from adult tissue into a youthful, embryonic-like state. These cardiac patches, when implanted into primates after heart injury, not only integrated into the host tissue, but began beating in rhythm with the heart, significantly restoring function. The success of this approach has led to approval for early-stage human trials, setting a precedent for regenerative approaches that could eventually replace stents, bypass surgery, or mechanical support devices with living, self-repairing tissue. This same paradigm is being applied across a wide range of organs. In the liver, researchers are using iPSCs and organoids, miniature, simplified versions of organs grown in vitro, to model diseases and screen therapies. But more importantly, these liver organoids are now being tested as therapeutic implants to replace damaged hepatic tissue. In kidneys, stem cell-based models are laying the groundwork for eventual tissue replacement or regeneration of nephrons, the filtering units lost in chronic kidney disease. In the brain, experimental cell therapies are showing promise for conditions like Parkinson's disease, where the transplantation of dopamine-producing neurons derived from stem cells can potentially restore motor function and cognition. The long-term vision is not merely organ repair, but functional rejuvenation. As people age, organs gradually lose their regenerative capacity, primarily due to stem cell exhaustion, senescent microenvironments, and accumulated damage. Regenerative medicine aims to overcome all three by replenishing tissues with new, healthy cells, often genetically matched to the patient, and, in some cases, genetically enhanced to be more resilient than the originals. This could mean replacing aged bone marrow to rejuvenate the immune system, implanting bioengineered pancreatic cells to restore insulin sensitivity, or delivering stem cell-derived muscle fibers to reverse sarcopenia. These are not speculative concepts. Many are already in preclinical development or early-phase clinical trials. Of course, the challenges are significant. Immune rejection remains a key concern, particularly with allogeneic, donor-derived cell therapies. Even autologous, self-derived stem cells, if reprogrammed improperly, can carry risks of tumor formation or genomic instability. The aging tissue microenvironment also poses obstacles. Inflammatory signals, fibrosis, and senescent cell accumulation can suppress the integration and survival of implanted cells. Overcoming these hurdles will require a systems-level approach, combining regenerative medicine with anti-inflammatory drugs, senolytics, and tissue engineering scaffolds that mimic the youthful extracellular matrix. However, as techniques improve, regenerative therapies will likely become increasingly modular, safe, and routine. Advances in 3D bioprinting and biomaterials will allow for the creation of custom tissue grafts designed to match the patient's anatomy and integrate seamlessly. Organoid transplantation may eventually extend to entire functional units, such as gut epithelium for inflammatory bowel disease or retinal patches for macular degeneration. For aging individuals, this could mean repairing organs not once but repeatedly over the course of a long, active life, using regenerative boosts to keep pace with biological decline. Looking further ahead, stem cell banking and tissue preservation could become integral parts of early adult healthcare. Young adults may store iPSCs or umbilical cord cells as future raw material for organ repair later in life. Insurance models may evolve to support these regenerative health reserves, offering people the means to repair rather than replace their organs as they age. The very notion of aging may shift from one of inevitable loss to one of periodic renewal. In the broader context of anti-aging medicine, regenerative repair will complement other strategies, pharmaceutical, genetic, behavioral, by addressing what no pill can, the structural and functional degradation of tissues. The promise is not just to slow the progression of heart failure, kidney disease, or neurodegeneration, but to restore function and extend independence, vitality, and dignity late into life. As clinical successes mount and public trust grows, regenerative therapies will become a core pillar of longevity medicine, perhaps even the most visible and inspiring symbol of how modern science can rewrite the rules of aging. In the near future, we may witness a healthcare model where a failing organ doesn't signal the beginning of decline, but the beginning of renewal. A new heart patch, a dose of neural stem cells, a printed kidney scaffold. These interventions may soon be as routine as hip replacements or cataract surgery today. With each advancement, the goal moves closer, not just to live longer, but to remain whole, strong, and capable across the full arc of a life well lived.